Hello, everyone. Thank you for joining us for the St. Croix Regional Session of Wisconsin Water Week. Welcome to Leveraging Intentional Partnership to Navigate Turbulent Waters, the St. Croix National Scenic Riverway and St. Croix River Association partnership. This presentation features 4 distinguished speakers, including Monica Zachay, Director of Programs at the St. Croix River Association, Dan Peterson, the St. Croix River District Interpreter for the National Park Service at the St. Croix National Scenic Riverway, Nicole Biaggi, Outdoor Educator at the St. Croix River Association, and Marion Schaefer, Biological Science Technician with the National Park Service at the St. Croix National Scenic Riverway. Each presenter will speak for 10 to 15 minutes, after which we will take questions from the audience. Please be sure to type your questions in the Q&A section and upvote questions you would like to see answered. Please join me in welcoming our presenters.
Thank you, Sophia. So as Sophia mentioned, I'm Monica Zachay, Director of Programs with the St. Croix River Association, and I get the honor of kicking it off today. So Nicole, if you'll go to the next slide. I'm going to start here giving a little bit of an overview of the St. Croix National Scenic Riverway and our partnerships. And I am actually seeing the second one. Can you go to the first slide, Nicole? Thanks, wonderful. So I'm going to give a little bit of an overview of the Riverway, and then I'm going to hand it off to Nicole and Dan to talk about our wonderful programs joint, and then Marion's going to join and talk about our invasive species. So for those of you who are not familiar with the National Scenic Riverway here, it is one of the very first, one of the first 8 designated federal wild and scenic rivers in 1968. At that time, Northern States Power, which we now know as Xcel Energy, gave a generous land donation to the Park Service, and at that time, Gaylord Nelson, governor for Wisconsin, as well, and the governor of Minnesota at that time as well. They passed, or they brought forth some language, and it became a National Scenic Riverway. So the boundary itself covers about 97,000 acres. The Park Service only owns about 20,000 of those acres. The rest are under scenic easement. And then it really includes just about 200 miles of protected riverway and a 7,760 square mile watershed. Next, please. Thank you. So one of the great things about, or several great things about the St. Croix National Scenic Riverway, outstanding biological diversity. Over 40 species of mussels in the riverway, as well as outstanding recreational opportunities, kayaking, canoeing, rock climbing, fishing, you name it. And it also has a great history and culture, from Native American history to the logging era that really shaped the watershed in the river. I wanted to talk a little bit about the River Association. So we were founded in 1911 and led by volunteers primarily. We had $1 dues back in the day. We are a membership organization. And in 2009, we hired our first executive director with a mission to protect, restore, and celebrate the St. Croix River and its watershed. And then in 2011, SCRA became the Friends Group of the St. Croix National Scenic Riverway. Since 2009, we have grown to a staff of about 10, so with a lot of different programming. And I'll talk about that next. So for programming areas, we have education and outreach. We do a lot of outdoor activities as well as K through 12 education, land protection, working with partners on conservation easements and fee title acquisition. We don't hold the land, but our partners do, and we help facilitate that. We have a pretty robust forestry program. Many of you maybe have heard of my St. Croix Woods. If not, go check it out. We really work with landowners and trying to match them up with the services for forest management and planning. We have habitat restoration, a water quality program, and the invasive species that you'll hear about more towards the end. And this presentation is really about the partnership. So like I said, we are the Friends Group of the St. Croix National Scenic Riverway. That means that, we have official agreements and we do a lot of joint programming and support. And especially during these turbulent times with COVID, it's come in very handy that a nonprofit, partner can really advocate on behalf of the National Park Service. We can act swiftly on behalf, of them. We do joint programming like our internship program, where the River Association, we will actually hire the interns and we'll put them out, in the Park Service or in the park boundaries with jobs to help them as the NPS budgets get cut and they're stretched pretty thin. Education and outreach, which Nicole and Dan will highlight, and then invasive species, which Marion will highlight. We also do habitat restoration. Arcola Bluffs is one of our favorites, just north of Stillwater, where we were able to get a Minnesota DNR grant to help the Park Service pay for the restoration of that property. And we do a lot of recreation and park use projects together, and however we can really support them. So with that, next slide. Little bit of a delay on that. Awesome. So that was just an introduction. And now I would love to turn it over to Daniel Peterson and Nicole Biaggi, who's just going to tell you about their work and the outstanding program that they've been working on over the past year or so. Thank you.
Great. Well, thank you so much, Monica. Yeah, so this is Dan Peterson. I'm with the National Park Service in Croix National Scenic Riverway, and one of a great partnership that we have with the St. Croix River Association is an outreach program we call Rivers Are Alive. And it's a great way to get students and teachers immersed into this national park that we have, and also to learn about the watershed. And so Rivers Are Alive is, it's been a partnership for a number of years, and is really a way to tell students and schools that the rivers are alive, and they are alive throughout the seasons. And so this is a program that has a number of at least over 50 different types of programs that are all environmental education based, with curriculum, K through 12 programming, with science standards that meet to Wisconsin and Minnesota. But definitely it is a way to get kids into the riverways, but also into other habitats of the watershed. And so looking at the objectives of Rivers Are Alive is that really we want to make connections. We want to connect with the students, the teachers, of where they are in the region. Where do they see themselves within the watershed, or even within their community, making personal connections? How are they fitted into this watershed? If they live on a stream or a creek, that tributary eventually feeds to a river and then eventually feeds to the St. Croix and on to the Mississippi. How do they fit in that whole big puzzle? So definitely discovering their place in their watershed. And then the other aspect too is that we want to make them, the students, scientists, to learn about the biological communities that are found within the water, all the macroinvertebrates, to the fish, to the otters, to all the variety of life that exists within the river system. But then also as scientists to learn about the threats in the river. Next slide. So as I mentioned, is that Rivers Are Alive has been going on for a number of years. And every year, the National Park Service and the St. Croix River Association get together to plan out our year and it's become very popular. As an example, back in 2020, year 2020-2021 school year, we had 30 schools signed up, and with an estimate of about 6,000 students to be engaged in the Rivers Alive program. Until, as we all know, in March, April, and May of 2020, definitely the pandemic hit us. So that then led to canceled programs and programs in the spring and summer and fall. And it's like, well, gosh, what do you do in a pandemic? When you think about all the programs that we have, how do you make connections when now the connections have to be different? They can't be in person. So then the next question is, out of all the 50 programs we have, how do we convert these programs into a virtual experience? Well, we thought of, how about going online? And online, definitely a great portal place. But gosh, another question is, we have all these programs. Do we make all of the programs online, or we just pick a few of them? And so definitely, Nicole and myself, we chose 4 programs that were most popular within the Rivers Are Alive series. And we chose them thinking that, these would probably be more manageable in a virtual experience and probably more effective online. But once we get to that point, before even putting all of this content online, we have a bit of a menu to go through. So how steps to going online? Gosh, there is quite a bit of detail that goes into that, and one of which is hiring a videographer. Between the 2 organizations, we felt, well, we could probably do our own video of shooting a video of a ranger or one of the naturalists through the association to do some kind of guided program that would accompany the Rivers Are Alive online program. But we decided to go in with a professional videographer who would shoot the video and do all the editing and captioning required. So then it's coming with the outlines of all of our in-person programs and converting them into scripts for narrators to accompany into the videos. Once the videos are shot, then they're edited and reviewed and approved. Eventually it goes into publication. Well, and then we have to kind of step back too, is the development of a Rivers Are Alive online page. And so that page then has structure and showing a variety of videos that are in there. And once all of that is put together, then we have to think about promotion and marketing. And looking back into our records with all the schools that have participated through the years, it's going back to those schools and to those teachers and shooting out kind of a blast email of, hey, we've got a great program available for you, and it's available right in your classrooms, known as Rivers Are Alive Online. So at that point, we're developing live programs, scheduling of live programs. So yeah, there's a lot of work that goes into creating something like this online. In addition to keeping the website, is what we call Rivers Are Alive Online, keeping it updated with new content and new videos. But also thinking about, well, once we do go back into person, we've got a nice backdrop here, a great resource to have. Next slide. So here's a great view of Rivers Are Alive Online. You can go to the St. Croix River Association web page, and right there on the home page, you'll find a link for Rivers Are Alive Online. And so, as I had mentioned earlier, Nicole and I chose 3 programs that were most popular, and you can see them right below the virtual program there. Let's Go Mucking, Into the Weeds, and Let's Go Birding. The Junior Ranger program is something we just added 2 weeks ago. And so this, the website, Rivers Are Alive Online, very easy to navigate. It's a type of program that, so if for the Let's Go Mucking and Into the Weeds, Let's Go Birding, and the Junior Ranger all have their own set of videos and student resources and teacher resources. and that students can really just learn at their own pace. That's the neat thing about these programs too. And so while we have this page, we also provide an opportunity for students and classes to have live virtual classroom visits. So as an example, Let's Go Mucking, which is exploring the aquatic macroinvertebrates that live in the river. After going through 3 or 4 of the videos that are found within the Let's Go Mucking page and checking out the student resources, then the teacher can also sign up for a live Ranger Dan presentation, classroom presentation. So anyway, hopefully all of you can have an opportunity to visit Rivers Are Alive online. But I'm going to turn this, I'm going to turn it over to Nicole, and she's going to go into the weeds, if you will, about something else.
There we go. Sorry, I was muted. Thanks, Dan. So yeah, like Dan said, I will go into the weeds, into specifics about our Into the Weeds page, and we'll take a look at what just one program looks like. So as we made these different programs, Dan talked about how we choose which program we wanted to focus on. And then within that, our top 2 goals, I would say, were to create interactive and engaging videos that would get students participating with the video, not just watching, because there are so many videos out there online already that if we were going to create new videos, we wanted to figure out what we could do to make them unique and make them local and engaging. And then our other main goal was to make them very accessible. So, you know, the website is entirely free. You don't need to use anything to log in. You can just go to the website and anybody can look at the videos and access all of the resources right there. We also made it, you know, a goal to have closed captioning and audio descriptions for all of our videos. So this image here at the bottom is just a screenshot from our Into the Weeds page. So every page has a similar format with videos going down the page and text next to the video. There's typically an introduction, a couple body videos, and a conclusion video for each program. So this one is one of our body videos on the Into the Weeds page. And this is Ranger Sierra going on a hike at one of our local trails in Osceola, Wisconsin. And so she is going on the hike looking for invasive and native species. And she's taking with her a scavenger hunt sheet that we created that has native and invasive species on it, like a bingo sheet. And so students can then click this link right here next to the video and download or print out their own scavenger hunt sheet so they can follow along the video. And like Dan said, we were trying to pick programs that would be most effective. So this one seemed like it would work well online. That's why we chose this. And sort of the inspiration was Dora the Explorer. If anybody is familiar or can remember that show, it is a hike and the students are watching the screen and she's like, oh, do you see that over there? Look over there. And so that's kind of what this hike is like. Students can follow along with the scavenger hunt sheet and look for these species and notice them before Sierra does. And then they can take the sheet out. So she encourages them at the end of the video, now use this sheet on your own, you know, either with your classroom for the students who are in class or at your house to get outside and explore. And so next to each video is also this audio described version link, so students can click on that and go there as well. And then on our page, each page at the bottom has links to more resources. So there's student resources, which are made directly for students. So a couple examples for Into the Weeds student resources are here. There's the scavenger hunt that I just mentioned. So the sheet on the left side of the screen. And then in the middle, we have a reading. So a lot of our student resources include readings, activities such as a crossword or a word search, all different things that students can use to take the videos and the lesson further. We also have teacher resources. So this is a separate page that teachers can click on, and the resources are designed for teachers and written for teachers. So this one is called Math, Measuring, and Models for Into the Weeds, and it's designed for grades 3 through 8. So that takes the videos a little further because the videos are designed more for grades 3 through 5, whereas this can take it up a notch for some older grade levels if teachers want to. And so there's some background information for teachers to learn a little bit more about native species and what makes their strong root systems beneficial for our ecosystems. And then there's a couple pages missing that go along with this. I just put 2 pages here, but you can see, you know, if you're not familiar with super long native species roots and their benefits, you know, teachers can read about it and then they can see diagrams of those roots. And then with their students, they can actually use yarn and measure it to be the length of those roots, cut it off, and then stretch it across the classroom or stretch it outside and see how long the roots really are, because it's hard to imagine 15 feet of roots when you're just looking at the picture. And then they can compare it to non-native species like our Kentucky bluegrass and see the difference with their own eyes. And, you know, they get to measure, they get to use models. So it's a way to really take those videos further and extend the lesson. So in addition to teacher resources and student resources and those asynchronous videos, we also have live virtual classroom visits. And so these ones, teachers can sign up and schedule a time for their classroom to do a live program. And our programs right now are generally 50 minutes. We do them with Zoom or Google Meet. Teachers tend to like to use Google Meet the most, so that's the one we've been learning to use. And our most popular programs are Meet a Mussel for K2, Great Mussel Mystery for 3-5, Watershed and Jeopardy for 6-12. And all of these programs were originally in-person programs that were done in the classroom. So we have a lot of programs that we would do in the classroom when the weather wasn't so good. So in November or December, we might go to the classroom and do these programs. But we decided that they would work well as engaging programs to switch to be online. So what has made these programs successful, I have kind of listed down here. We do screen share some videos, which we found is successful on Google Meet, but not so much on Zoom. Lots of Q&A, so students get to speak up and answer questions. We get to ask them to make observations about the videos. They can make hypotheses and really participate vocally in the programs. And with our Great Mussel Mystery, there's a drawing part where students get to use a pen and paper to do a little drawing activity during the program. With Meet a Mussel and the Great Mussel Mystery, there's body movement. So students get to actually, use their hands to pretend that their hands are a mussel, and then they get to imagine what a mussel feels like using their hands and comparing that. Teachers have told us that putting vocabulary on the screen is really important, so we have been doing that. And then with our Meet a Mussel, we've tried singing, which has been fun for me, and I think the students too. It's a little bit challenging if they are coming in on their own devices versus being in the classroom all together, just having so many different students on their own devices trying to sing at the same time. But we've been trying different things, and some of them need some tweaks, but it's all fun. And then competition and challenges are really great too. So our Watershed and Jeopardy is actually a game of Jeopardy. And so the first 20 minutes of the program, this picture here is Jeremiah and Katie. Jeremiah is leading right now. He's talking about aquatic invasive species for 20 minutes. And then Katie will jump up and then she'll do a game of Jeopardy and see what the students can remember about the aquatic invasive species through a fun game where the class kind of gets split in half and they actually compete for points. And it's really effective. And so these programs have worked well for us, and we hope to continue offering them going forward after, we get through this COVID period. So lastly, I want to wrap up with challenges and successes that we have found. So a major challenge is that Dan and I actually started with, the Riverway in about June of 2020. So less than a year ago, which was kind of right in the midst of COVID. And it was a cramped timeline for us to get started and to get these things rolling before the school year started. And our goal was to get these online resources available by September, which ended up being about October. But the other issue that we faced all year, and it's an issue of trying to get things done immediately for that very season. So seasonal changes. With Into the Weeds, we started producing those videos in August, which is when there's goldenrod and New England aster and really tall, beautiful, colorful flowers to identify. And then the video, by the time that it was edited and produced and promoted and got online, it was October and those plants weren't there anymore. So it was a little bit late. And then the same thing happened with the winter. We wanted to get some snowy videos out there with winter related material. By the time that the snow landed, we did the videos and got them out there. Now we we got the video out there 2 weeks ago and now the snow is gone. However, luckily these videos will be relevant next year. So we will have them fit with the seasons in the coming year. It was just hard to do, trying to get them created and out there for the same season that you're filming them in. Communication and coordination is always a challenge between, two separate offices, two separate groups. But during COVID has been especially challenging. We've just been having to do a lot of, meetings, weekly meetings, Dan and I, to make that happen. The other challenge is that it's difficult to gauge success. So we made this self-reporting form to try to pull in numbers for grant reporting from teachers. And it's really simple. It just asks for a couple pieces of information, but it just hasn't really been successful. We haven't found that it's working to get teachers to fill it out yet. And then our feedback survey also hasn't really received a lot of response yet. So we are still working on figuring that out. And then we've had some difficulty with response from teachers just because they're overwhelmed. And overall, just trying to work on building up our program and getting teachers engaged with it. And then lastly, I'll just mention our successes, which we've had, they're interactive and engaging, and the topics are not commonly offered. So freshwater mussels and aquatic invasive species is something unique that teachers are looking for, and they're happy to try something different. We reach schools within our watershed and beyond, and we're hoping that going forward, we can use these online resources as a pre-activity for in-person field trips. So going forward, we just have to figure out how to transition these resources into our traditional education model and how to bring them both together. And that is everything that we have to present about our Rivers Are Alive program. So I'm going to hand it over to Marion, who's going to talk about aquatic invasive species next.
Awesome. Thank you, Dan and Nicole. I want to do the scavenger hunt. I'm just going to get mine shared here. Okay. So I'm Marian Schaefer. I'm with the National Park Service at the Riverway, and I'm lead of the Aquatic Resources Program. And much of my work actually focuses on AIS monitoring, though I oversee and protect and preserve all aquatic flora and fauna. Katie Sickman, she's one of our many amazing partners at SCRA, and she's SCRA's invasive species coordinator. She's very heavily involved with the project and is the lead on many of the education events I'll be highlighting today, but Katie's unable to co-present with me as she's leading a session later today on our winter emerald ash borer monitoring within the watershed, and her talk's at 1:15, so you should check it out. So as the Friends Group for the St. Croix National Scenic Riverway, SCRA supports our work within the watershed and our overall mission, and that's to protect, restore, and celebrate the Riverway. And by working together, we endeavor to raise awareness of aquatic invasive species, and here on I'll just abbreviate that with AIS, and also to combat AIS-related issues within the basin. Like water bodies all over the world, the St. Croix and its tributaries are threatened by the invasion of invasive species. And with the St. Croix being one of the cleanest tributaries to the Mississippi River, it's crucial that we try to combat this as early as we can. It's a very high value fishery, and a diverse, really healthy ecosystem includes at least 40 native species of mussels, which have been here actually since prior to European settlement. And several of these mussels are rare or endangered. And with the introduction of AIS into this system that's very complex, they really threaten the ecological integrity of the Riverway, and then also our unique cultural resources and our outdoor heritage. So in an effort to mitigate any impairment caused to the ecosystem by AIS, together with partners, SCRA in 2014 developed the St. Croix Basin Aquatic Invasive Species Strategic Plan. And this has been developed over the last several years and will continue to develop as we learn more and we have new threats on the horizon. Overall, though, the strategic plan overarching mission is to prevent, contain, and control AIS in the St. Croix River Basin. And there's 4 main goals of this plan, and those include preventing the introduction and dispersal of AIS into the St. Croix River Basin water bodies. Also to contain AIS infestations through early detection and rapid response, and by fostering good communication and knowledge among partners, and also educating the public. Another goal is to control and eradicate AIS infestations that are already present in the watershed and prevent them from spreading further. And finally, it's a goal to ensure that the strategic plan is being implemented and to continue to monitor the plan for its effectiveness and success. So some of the main threats that we have currently for AIS include purple loosestrife and yellow iris, Eurasian water milfoil and curlyleaf pondweed, and also zebra mussels and bighead and silver carp, which are on the horizon. Those are just a few, there's many others. So most of these introductions of these species are a direct result of our own human activities. And so yellow iris and purple loosestrife are very beautiful plants. And so they were commonly planted actually intentionally along the shorelines in the upper St. Croix Riverway as kind of decorative plants. And so what happened is they escaped and spread, and now they line parts of the riverway, the whole riverway, suffocating out really important native plants like wild rice. Asian carp species, as we all know, have made their way up the Mississippi River and now are getting closer and closer to the St. Croix River. And actually today, bighead carp have been caught in the St. Croix, in Lake St. Croix, and also silver carp have been caught near the mouth of the St. Croix. Zebra mussels are one of the worst and most heavily infested right now, and we're seeing their ranges expanding fast and furiously. And what they do is they directly outcompete for habitat and food resources with native mussels, and they can actually displace endangered native mussel species such as the winged maple leaf. So the potential impacts of all these invasive species are significant, and they are widespread, ranging from ecological to economic to cultural. And with the watershed being approximately 7,800 square miles, it's essential to collaborate with partners such as SCRA and others to help prevent the introduction and dispersal of AIS, and then also to try to contain and eradicate infestations if possible, because once they get too out of control, there's just it's not humanly possible to get rid of them. So we want to stop them before that happens. So the zebra mussel, we'll talk a little bit about the zebra mussel, our favorite. It found its way into the St. Croix River back in 1995, but it wasn't discovered to be reproducing in the river until 2000. And so to monitor and better understand the populations within the river, we partnered with SCRA to measure zebra mussel densities at 10 different locations along the lower riverway. And at each location, we combined 3 standard cinder blocks that are enclosed in a wire mesh caging to protect them. And they're deployed usually before mid-April and then collected in early October of each year. And then they're also affixed with these little tiny hole loggers, which track temperature and light intensity over time. And then also, in addition to these cinder blocks for early detection purposes, and just as a general indicator of general presence or absence in the upper riverway in the Namakagon, we also deploy submerged zebra mussel settling plates, they're called, and we usually put these right next to the impoundments on the Namakagon River just to see, are they there yet? And if not, great, but they haven't been, but we need to keep monitoring that just to be sure. And pictured here in the middle is a picture I took last summer, or last fall rather, it was in the lower riverway at a site located near the confluence with the Mississippi. And this was by far the most heavily infested block. As you can see, it took a very long time to count them, and it was an estimate, obviously. But if anyone can guess how many zebra mussels are on that block, they win a free, fresh, delicious seafood boiled dinner. Additionally, so we also conduct, together with SCRA, some early detection monitoring through a combination of qualitative and quantitative methods. And we're looking at zebra mussel babies or their veligers. And we do this sampling in lakes in the whole St. Croix River Basin. And the need for these early detection monitoring practices has risen with infestations in watershed lakes, such as Big Mackenzie Lake and Deer Lake. And though we haven't seen any new infestations, it's crucial that we continue to monitor these lakes within the watershed annually to make sure they're not spreading. So in 2020, we sampled veligers using 2 different methods. Our quantitative sampling was done via handheld pumps, and this occurred in 7 different river locations along the Riverway all the way from St. Croix Falls down to Prescott. And then we conduct our qualitative sampling using plankton tow nets that we tow horizontally in the water column. And we did this on 30 different lakes, equaling over 27,000 surface water acres throughout the watershed. And luckily, no new populations were found. And this past year, we also started sampling for a new aggressive critter. I mean, not new to the nation, but just a new method of sampling. We started sampling for the spiny water flea, and in total, we sampled 20 lakes in Pine and Chisago counties, and the results showed that there were no spiny water fleas detected. So here are just some maps to kind of clarify what I'm talking about. On the left-hand map, the blue points show our veliger tow locations within the watershed, and the orange points on the upper right show the zebra mussel early detection plate samplers. And in 2020, we deployed samplers, again, May through October, at 4 locations, and these included the Namacoggin, Hayward, Trigo, and Gordon dams. And there were no zebra mussels found attached to these samplers. Actually, not really anything was attached to them, oddly. I think there's one tiny baby native snail species on one plate. So there was not much going on up there. The red points show cinder blocks, which are better shown in the right-hand map. So there it shows the 4 pools of Lake St. Croix and then the 10 sampling sites for the cinder blocks within that area. In 2020, we sampled these 10 locations from right above the Stillwater Lift Bridge down to the Mississippi confluence. And after we retrieved the blocks, we examined them immediately, and all zebra mussels are counted. And if possible, specimens are removed and preserved for future size determination, age class, structure, and possible genetic analysis. And 2020 results just confirmed a continual increase in spread and densities across all sites. And when you compare across previous years, the numbers are just continuing to rise. So in addition, we do another method of quote unquote sampling, and this is called our dry dock boat counts. And this is done at there's a dozen marinas that operate on the lower St. Croix River, and several are visited every fall to examine boats that have been pulled to be dry docked for the winter, and usually examine about 100 boats in each marina for the presence of attached mussels, and then we'll do counts and age structure classification. And then we kind of just use this as another method of indication of the population level over time. And so the marina slip boats really offer the perfect substrate for mussels, especially for like post-settling animals, post-veliger state, because many of the boats remain at the docks all season. They have dark little protected spaces for settlement, and then since they're in that slack water near the surface area, they really enjoy this area. And so it's a really unique, it's funny to call the sampling method, but it provides it allows us to assess relative abundance along the infested portion of Lake St. Croix. So, and in this past November, we visited dry dock boats at 9 marinas from north of Stillwater down to Prescott, and this was a total area of approximately 25 miles that we covered in one day. Some good data. And so as in past years, the marinas that had boats docked in the upstream portion of Lake St. Croix tended to have less boats with 1 or more animals attached, while boats that were moored downstream of the Hudson, Wisconsin Narrows had a greater than 50% chance of being pulled from the water infested. And this reflects a trend from previous years that continues to suggest that there's within pool reproduction and recruitment happening with these animals. We actually have been doing these dry dock boat counts since 2000 and will continue to do so. One interesting note is that when you look at peaks over the last 20 years with respect to river densities on known substrate or the river bottom, these numbers are reflected in the number of zebra mussels attached to the boats. And so it's just further evidence that long-term monitoring of boats has been a valuable and very indicative tool. So kind of switching to plants a little. NPS also partners with SCRA to monitor the St. Croix Namakagon for yellow iris and purple loosestrife. These are both relatively new efforts, but they've already both proven to be really effective in controlling the populations and the spread. So the yellow iris is really rapidly expanding its range along the Riverway, and today it's found throughout the headwaters region, as well as all the way from St. Croix Falls to Marine on St. Croix. It's very easy to identify when it's in bloom, like after the flowers mature into seed pods. However, it's often mistaken for native blue flag iris. And so in 2020, AIS interns and staff monitored a total of 91 miles of river, and this included 39 miles of the Snake River to inventory the presence of yellow iris. And these maps show, the yellow line shows the pathway that was inventoried. This whole section, the entire section, and the yellow dot, I'm sorry, the red dots show yellow iris populations, and the blue show the blue flag iris populations. So monitoring and removal efforts for the yellow iris began back in 2017, and have been ongoing each year since. And because of this, now today, monitoring efforts have completed the entire St. Croix Namakagon rivers, which is very impressive. Without this joint effort on behalf of NPS and SCRA, this would not have been possible. And without a doubt, like populations by now would have spread and infested the Riverway beyond our control. Like I said, it would have been, we would never see the end of yellow iris. So it's been very effective. Also in 2020, this is a new thing we started doing. We began monitoring and managing for purple loosestrife. 3 sections of the Riverway were monitored, and this covered a total of 39 river miles. And so during these outings, the plants are removed by cutting at the base of the plant before seed dispersal, so you're not spreading the plants even more. And the plants, we just put them in large garbage bags and disposed of them responsibly. And then, of course, GPS located every single plant on the Riverway. Sections of the Namakagon near the Phipps flowage that were heavily infested were treated with herbicide treatment to ensure control, and approximately 1 acre total was treated with herbicide. So we are learning a lot through our efforts and continue to modify our techniques and methods to increase success. And so this is really exciting. In 2021, the beetles started jumping too fast. In 2021, SCRA will be raising Galrucella beetles to be released as a purple loosestrife biological control agent. They have been proven safe and effective, and they are likely the best long-term control method for loosestrife. This will negate having to use expensive herbicides or higher cost treatments. Also, exciting news is that NPS recently submitted a new grant application for a fiscal year 23 large-scale removal of purple loosestrife and yellow iris, and this is going to be kind of like a really extensive, large-scale effort that will kind of hit everything heavy in 1 year and hopefully try to eradicate these populations entirely. Shifting gears a little bit to some of the citizen science opportunities. SCRA provides a lot of unique opportunities for folks in the area. Project RED, which stands for Riverine Early Detection, is a neat program. It's a monitoring program that teaches participants what invasive species threaten local rivers and how to differentiate them from the native looking ones, and how to keep an eye out for them, whether they're in their kayak or canoe or walking on foot, just hiking. It really helps even people that are in my field, biologists. It's hard sometimes to differentiate these, so you really have to look at key identification features to know the difference sometimes. So this online virtual training in 2020 reached 13 participants. In addition to that, there's an algae alert network, or an algae alert program, it's called, where volunteers team up with SCRA, NPS, and the US Geological Survey to monitor the water all the way up and down the St. Croix River for toxic blue-green algae. And this volunteer monitoring actually helps researchers with USGS better understand if, when, and where toxic algal blooms will occur along the St. Croix. And with climate change, it is being foreseen that these blooms will get worse and worse in the future. And so this is actually work is and being enveloped into some really high scale research projects that the USGS is doing. And in 2020, sampling was done at 8 sites with the USGS along the St. Croix to assess the algal blooms and kind of track their distribution over time from late summer to early fall. And in total, they had 9 volunteers that collected a total of 56 samples.
Marion, thank you so much for your presentation. I'm afraid we're already
Oh, no.
out of
Okay.
time. No
That's
worries.
okay. That's okay.
We've had a lot of fun. I just want to make sure
Yeah.
everyone can get on to their next presentation. I
There we go.
don't see any questions in the chat. If anyone has any questions, please feel free to continue to enter them and we'll make sure that our presenters answer them. I want to thank Monica, Dan, Nicole, and Marion for their amazing presentation. And we have now come to the end of our session, but if you would like to learn more about the AIS work being done in the watershed, including emerald ash borer monitoring and citizen science opportunities, please join us at the St. Croix River Association event tomorrow from 1 to 3 p.m. Details are available on the conference website. Please note that you have a short break now to visit the exhibitor and sponsor booths, network, and participate in the Wisconsin Water Week challenges from 11:15 to 11:30. Your next session will be Charting a Path for a Socially Regenerative Agriculture with Adam Reimer at 11:30 a.m. Thank you all and have a wonderful rest of your day and conference.